Hey folks, it's me, Mr. Lang, your friendly neighborhood AP biology teacher, and today I'd like to talk to you about cellular respiration, which includes the electron transport chain, which should sound familiar, and also ATP, which should sound familiar as well, because you need that stuff. The real question is, how do we go from this delicious-looking array of foods to this, which is not delicious looking, but is very important, and if you haven't figured it out yet, it is adenosine triphosphate, or ATP. Where is this happening? Where do we take that food and convert it to ATP? Well, that's the mitochondria, or as you all like to say, the powerhouse of the cell. But here's why it's the powerhouse, and this is why it's important for you to understand a little more than just regurgitating that it is the powerhouse. So here's our mitochondria. Here's our outer membrane. Here is an inner membrane, so there's an intermembrane space here. And there's also this fluid-filled area here that's called the matrix. And I remember the matrix because I think about Keanu Reeves swimming through the inside of the mitochondria. That reference might be too old for you, a little dated, but it works for me, so use it if it works. Here is a broad overview of everything that's going on. We've got three main steps. We have this step called glycolysis. That's where we're going to break glucose down into two pyruvates. Those are going to go into the Krebs cycle. The Krebs cycle is going to result in us getting some ATP. Oh, sorry. Glycolysis also results in a little bit of ATP. But the real win here is that we're going to get electrons that are carried by some very familiar looking characters, NADH and FADH2, to the electron transport chain, which is going to drive ATP synthase, which should sound very familiar based on what we talked about earlier on in the year with photosynthesis. So here we go, a little bit of a deeper dive into each of those different ideas. So glycolysis is happening in the cytoplasm, so just outside of mitochondria. We will take glucose and we will have an input of two ATPs. So we're going to use two ATPs, which is going to generate two ADPs that can then go get recharged. And again, College Board doesn't care so much about the intermediary steps, but they do care that you know that there's a couple of pyruvate molecules coming out. So we're going to break a 6-carbon glucose into two 3-carbon pyruvate molecules. And in the process of doing that, we are going to capture some energy in the form of ATP and NADH. So we're reducing the NADP plus by adding some electrons, and we're going to charge up some ADPs to ATPs. We're going to make 4 ATPs overall, which offsets the use of ATP and makes this sort of an energy lucrative proposition for us, because we're going to end up with 2 more ATPs than we started with. And we're also going to make some NADHs that are going to end up going to the electron transport chain. So citric acid cycle, also known as the Krebs cycle, pyruvate comes in, gets combined with acetyl coenzyme A. As we go through this process, the really important part here isn't necessarily the intermediary steps. It's probably important that you know that acetyl coenzyme A combines with pyruvate to form citric acid, but then from there on, basically, you want to know that we're going to be releasing some CO2, and we are going to be generating NADH from NAD+. It's that electron acceptor, and the electron acceptor is going to be passing directly along to the electron transport chain. We're also going to make a little bit of ATP from ADP and an organic phosphate, but again, the most important thing here is that we are taking NAD+ and creating NADH and FAD and making FADH2 for the electron transport chain. Now, the electron transport chain, with the subtraction of the sun being important, which it was in our last iteration, it's going to look sort of similar, right? So we've got let me move myself out of the way here. I'm not sure where to put myself. There we go. So we've got some NADH and FADH2s being broken down. Those electrons that they were carrying are being moved through various compounds that are the electron transport chain. As we're doing that, these protein pumps, these integral membrane protein pumps, are using some of that energy to pump hydrogen ions into the intermembrane space. Remember, we have that space in between the outer and inner membranes. This is the matrix here, so this is where Krebs cycle is happening. So this is where we're generating that NADH and FADH2. So we're going to be pumping hydrogen ions using that energy. And then at the end of this, we've got this excellent little electron acceptor that is very, very electronegative, so it really has a strong pull on electrons. And we will end up using those electrons to attract some hydrogens, and then we'll make some H2O, and that is known as metabolic water, and we generally will either use that or filter it out of our blood, and after diffuses from our cells, and get rid of it as urine, or we may need it because maybe we're not drinking enough water. So in any case, this should look really, really familiar. These pumps are creating a proton gradient out here. Concentration of protons is higher out here than it is in here. That allows for ATP synthase to use the cascade of protons to charge ADP into ATP, and that is a big deal. As long as there's oxygen here, this will happen, and we will make lots of ATP from every glucose. If this oxygen isn't here, this whole thing starts to shut down, because if I don't have a greater concentration of hydrogen ions out here than I have in here, flow is going to slow down, and we're not going to make as many ATPs. So the overall efficiency per glucose molecule of the electron transport chain is completely dependent upon the availability of oxygen within your cells and the accessibility that your mitochondria have to the oxygen within your cells. So overall, we've got glucose and oxygen coming together with carbon dioxide, water, and ATP. And the ATPs that we can make overall, it looks, I went a little too far, looks like this. So in glycolysis, we're going to make 2 ATPs. In the Krebs cycle, we're going to create 2 ATPs, one for each of the pyruvate molecules that go in, and we're going to make about 34, max of 34. So that's if enough oxygen is available. So that's an important if right there, because anything less than optimal oxygenation is going to result in us not making 34 ATPs per glucose molecule. But in optimal circumstances, for 1 molecule of glucose, we can make 38 molecules of ATP, or we can charge up 38 ADPs into ATPs if you want to think about it that way. And that's great, because that's the cellular currency that we use to drive every reaction in our body, as you already know. Okay, what if there isn't any oxygen around? Well, if there's not any oxygen around, glycolysis will happen just like usual. Basically, what we're going to do is we're going to go through a couple of forms of anaerobic. An means without, so without oxygen cellular respiration. So glucose is going to be broken down into pyruvate, and that's where similarities go away. There's no Krebs cycle. And actually, in some organisms, there can be an electron transport chain, but the final electron acceptor is a sulfate, which is one of those polyatomic ions from chemistry, and it's not as electronegative as oxygen, so it doesn't have the same pull, and it's not as efficient. But these are the other 2 anaerobic respiration ideas. So one is alcoholic fermentation, that's ethanol, alcoholic fermentation, and lactic acid fermentation. So so basically what happens is in different organisms and for different reasons, us eukaryotes in general and some bacteria go through lactic acid fermentation. Without lactic acid fermentation, you wouldn't have any yogurt or cheese, so thanks bacteria, and thanks for anaerobic conditions. And that is going to happen in your muscles as well and other cells. If you don't have enough oxygen, you're going to start creating lactic acid. Alternatively, if we were talking about all yeasts and some other bacteria, we're going to end up with lactic acid fermentation, which creates CO2 as a byproduct and also ethanol as a byproduct. So, and they're going to allow the production of some ATPs, just not as many. So anaerobic respiration is not nearly as efficient in ATPs produced per glucose, but it does allow us to still break down the foods that we eat into glucose and allows us to create some energy that's usable at the cellular level in the form of ATP. So that's an important thing as well. Alcoholic fermentation, that's responsible for things like beer and wine, and pretty importantly for most of us, bread wouldn't rise unless we had that yeast creating the CO2, and it also does create some alcohol that it sort of evaporates out as the bread is baking. So that is sort of a basic primer on cellular respiration, both aerobic and anaerobic. So all the stuff that I talked about before this slide would be considered aerobic or aerobic cellular respiration because we have an ample supply of oxygen. Here, this will only happen if we have little or no oxygen present to act as that final electron acceptor in the electron transport chain, which ends up causing us to not have the ability to create the proton gradient that we really needed here to make ATP synthase spin and make and make all of the ATPs that we needed to make. So I think that's all I wanted to say. If you have any questions, make sure you stop by and see me in class, and we'll talk about this, and I can buzz through the slideshow again. We can talk about the ins and outs. Have a great day. Be safe and make good choices.